Hi, my name is Zoltán Egyed. Welcome to our event organized by the Innovators Club of Silicon Valley. We are a non-political association of people involved in innovation. You can visit our website at hungariansv.com. Our presentations during normal times are usually at SRI International in Menlo Park, California. During the pandemic, we did our presentations online only, and we are about to return to presentations in person. Our speaker today is Daniel Bence. He will be speaking about his exploration of immunology. I'll be muting participants during the presentation. When it is time for the questions, please remember to unmute yourself when you are asking a question. I can mute you, I cannot unmute you. Please help me to welcome Daniel Bence.
Okay, yeah, it took me some time to finally unmute myself. Yeah, thank you very much for the invitation. It is a pleasure to be here talking about our research. So what I'm going to do today is I'm going to try to give you an overview, if I'm going to be able to move my slides, about the immune system, how the immune system works in disease and in health. And we will get into T-cell mediated immunity in acute and chronic viral infections, more specifically. Our lab is interested in the immune system in general, but today I decided to only go with T-cells and work that covers T-cell mediated immunity. We will look at what happens when T-cells can clear a pathogen efficiently and what happens if they fail. And we will look at what happens to T-cells when they undergo exhaustion and how do we study this phenomenon in a lab. And during this whole presentation, I'm going to show you different technologies that we are using in the lab to understand T-cells and how they work. And this will include single-cell technologies that are cutting-edge new technologies recently developed, but also other technologies that can map, for example, 3D genome conformation. And I'm going to talk about these in detail. So what do we do in the lab? What does an immunologist do in the lab? So we are studying the immune system, all the cells in health and disease, which includes viral infections, cancer, and autoimmunity, and many other diseases, obviously. The immune system is working in basically two settings. One of the arms is the innate immune arm of the immune system. The other one is the adaptive arm of the immune system. Innate immune cells, for example, like macrophages, dendritic cells, or natural killer cells, don't need any additional help. These cells patrol all over the body. They look for pathogens, cancer cells, and they very efficiently find them and kill them. But if they feel that they need some extra help, they can also activate the adaptive immune system, which includes T cells and B cells. And as I referred to this previously, today I'm going to talk about mainly about T-cell mediated immunity. So how do these two arms work together to clear an infection? Let's look at an example. So when there is a viral infection or cancer in the body, macrophages and dendritic cells can sense these problems, and they can basically take up some parts of these pathogens, take up one part of the parts of the virus or parts of the cancer cell, and these will serve as antigens. So antigen can be anything that triggers an immune response. So these cells not only attack these pathogens, but also take the parts of these pathogens to lymphoid organs, for example, the lymph node. And once they get into the lymph node, they activate T cells by presenting them these pathogen-derived molecules. this will activate T cells and B cells. And these cells, once they are activated and multiplied in number, can travel to distant organs, tissues, and they will eliminate the pathogens. So how do these T cells recognize the antigen? Here we basically just want to zoom at the interface of dendritic cell T cell interaction. And what you can see here is that when dendritic cells activate T cells, they present the antigen, this red ball on this major histocompatibility complex. This is a protein complex that can present antigens to T cells. So dendritic cells present the antigen on this complex, and the T cell will be able to recognize this by its Tcell receptor on the cell surface. If the T cell recognizes the antigen, it will undergo activation and expansion. So the T cell multiplies in number, it starts to divide, and then it migrates to peripheral tissues. And this is the effector phase. When the T cell go into the tissue, it finds the tumor cell or virally infected cell, and it's going to recognize the antigen on the cell surface of the tumor cell or the virally infected cell. And the T cell is going to kill it by different mechanisms, some of which includes these cytotoxic molecules, granzyme B or perforin. The T cell is basically injecting these molecules into the tumor cell, and this will basically blow up the tumor cell. So this is a really harsh method, but it works very well. So how many antigen types can our T cell population recognize in our body? The short answer is all the possible pathogens. This is kind of a crazy thing, because if you imagine, you can imagine that there are multiple different types of pathogens all over us and like all around us. And there must be a very, very efficient mechanism that the T cells basically use to recognize all of these and defend our body against all of these pathogens. And the answer lies in the T cell receptor. The T cell receptor is on the cell surface of T cells. It's a dimer. It's a protein dimer. It is made up of two chains, alpha chain and beta chain. And the question is, how is it going to be so flexible to recognize virtually any type of pathogen? Now, to understand this, we have to look into how the T cell receptor is made in a T cell. So there is the beta chain, and first the beta chain needs to be formed and synthesized, and it happens at the DNA level. So this is the DNA of a T cell that encodes the genes for the beta chain. You can see that there are multiple gene segments that needs to be basically combined in a random fashion, in a kind of random fashion. And there is this process called recombination, which basically recombines these gene regions. And this will lead to a functional T cell receptor gene that can be transcribed. So transcription is the process when you make mRNA from the DNA, and the RNA can be then translated into protein. So first, the T cell is going to make this beta chain of the T cell receptor, and then it's going to use a very similar mechanism to make the alpha chain. And this process is a random process, so you are mixing up these different gene segments, and it's imperfect. So this imperfect way of assembling the TCR receptor is making the system kind of perfect, because this is the way how the T cell repertoire is going to be so broad, it's going to be able to recognize basically any type of pathogen. So it is estimated that the TCR repertoire of an individual is around 10 to the 19. So we have 10 to the 19 different T cells in our body with different T cell receptors. And you just need to select the right ones to clear the infection or cancer. So let's look at a temporal view of how T cells work in an acute infection. So once the infection is established, these immune sentinels, dendritic cells, macrophages, as I referred to this previously, will sense the virus, and they will travel to lymphoid organs and activate T cells. T cells will undergo clonal expansion. So only those T cells will be activated, and only those cells will be only those T cells will be multiplied that are capable of recognizing the pathogen. This is what I mean by clonal expansion. So once the T cells are multiplied in number, they will be able to clear the virus or the pathogen. And then at the end of this process, you're basically generating a very efficient memory response, which means that the T cells are going to leave, they basically die, and only some of these clonally expanded T cells will make it to become memory T cells. A very similar thing happens when we vaccinate ourselves. That also induces clonal expansion of T cells. Obviously, the pathogen is not in the body in that case, but the right T cell clones will be activated, and those T cell clones will be able to form memory. And during pathogen re-encounter, when the body again faces with the pathogen again, these T cells will be really efficient in expanding and killing the virus, for example. So this is a really ideal case, and it works most of the time. But what happens if T cell basically fails to clear the pathogen? So here we look at an example where there is an infection or cancer, which the T cells or the immune system cannot fight off. So here, these cells, the sentinel cells, macrophages and dendritic cells, can although sense the infection and the cancer cells, but once they activate the T cells, they fail for some reason. They cannot efficiently clear the virus, they cannot kill the cancer cells. So what happens is that they continue to expand, and there are more and more T cells that are not so efficient. And some of these T cells, due to the fact that they've spent so much time in this harsh antigen-rich environment, they undergo T cell exhaustion, which means that they lose their function and they cannot clear the pathogen. And this will go on, and this leads to a pathogen-host stalemate. That's how we call it. And the problem with this is that it gives more time for the pathogen to evolve and being able to basically evade the immune system on the long run. And this is a really big problem with cancer. So let's talk about T cell exhaustion a little bit and how exhausted T cells look like. So as I referred to this, T cell exhaustion occurs in cancer and chronic viral infections. Chronic viral infections include HIV or hepatitis C, for example. And the problem with this is that the pathogen is there for so long time that it will lead to chronic activation of the T cell through its TCR. And this chronic activation leads to problems within the cell, like transcriptional programs that can actually lock the cell in the exhausted state, and the cell cannot function properly. So some of these genetic programs include co-inhibitory receptors that are made, and the cell is putting these co-inhibitory receptors on its surface. And this will dampen the T cell mediated immune response. The other problem is that as a result of these co-inhibitory receptors, the T cell will lose its cytotoxic functions. So it cannot efficiently kill the target cell. The other problem is the lack of proliferative capacity of the T cell, which means that the T cell cannot fully reproduce itself. There are going to be persisting T cells, which can stay in the cancer tissue for a long time, but these cells are not functionally efficient, and therefore they just fail to clear the virus or cancer. So there are ongoing therapies, or there are actually FDA-approved therapies to block some of these co-inhibitory receptors already with antibodies, and these are really successful in the clinic. So these immunotherapies are definitely the future. The other immunotherapy application is to isolate T cells from a patient's blood and then engineer these T cells and reinfuse them into the patient to kill the cancer cells. The problem with this is, again, exhaustion, because once you reinfuse these cells, they will get into the tumor tissue and they again undergo exhaustion and they cannot efficiently kill the tumor. So we definitely need to further understand what's going on when the T cells undergo exhaustion. And that's what we are doing in the lab. So here I'm going to show you some of the results that we have from a mouse model where we can induce T cell exhaustion in the context of a viral infection. So here we are using two different viral strains, LCMV clone 13 and LCMV Armstrong. One of them, the Armstrong virus, induces an acute infection, which means that the mouse can easily fight it off within approximately 20 days. The other one, clone 13, cannot be cleared. So this virus will induce chronic viral infection in the mice that can last up until day 80 or 90. So the virus is going to be present for a long time. And in this infection model, we can see exhausted T cells, which are really similar to the exhausted T cells that we find in human tumors. So once we did all these infections, we can isolate different organs. And then after dissociating the organs, we can look at antigen-specific T cells that are reacting to the virus. And then we can do single-cell RNA sequencing coupled with single-cell TCR sequencing to understand how individual T cell clones react to the virus. So let's look into how single-cell RNA-seq works. So once you have your T cells isolated from this infection model, you can load them into this 10x Chromium controller. This is a microfluidics device. So you add your cells and you add beads that contain DNA adapter sequences. You also add to this reaction some oil and obviously some of the other compounds that are needed for the reaction, enzymes, and so on and so on. So at the end, what you get is a droplet. And in a droplet, you will have one bead and one cell. You can lyse your cell in this droplet and you release the mRNA content of the cell, which is basically a transcript of what genes are active in this cell. So now the mRNA has a poly A tail on it. So all of the mRNAs that are produced in a cell are having an mRNA and having a polyadenylated end. And these poly A tails can be used to attach these mRNA sequences to the beads. So on the bead, as I said previously, there's a DNA sequence, which has a poly T end. And then your mRNA from the cells are going to attach to this poly T fragment. And then you can do reverse transcription, which means that you are converting your RNA into DNA. Why is this important? Because we can only do DNA sequencing. So at the end, you want to sequence your DNA. And then at the end, after sequencing, you're basically ending up having a count matrix. How many mRNAs you detected for one gene and what cell it relates to, and also the TCR gene information. So having all this information from single cells, thousands of single cells, will allow us to look at heterogeneity in the sample, to look at all the different cell types that are present in our sample. And this is an atlas that we generated from T-cell exhaustion by looking at CD8 T cells specifically. So on this map, this is called a UMAP. We are presenting more than 96,000 T cells from chronic viral infection. And you can see that there is a lot of heterogeneity. So there are multiple T cell subsets that we can detect with this technology. There are many of these subsets that were previously unknown. So now we can follow up these observations by more specific investigations to look at the function of some of these T cell subsets. But how do we define these T cell subsets? So here on this heat map, you can see gene expression profiles, which means that we are presenting what genes do these different T cell subsets express or use. You can see that, for example, in naive T cells, which are these purple ones, you can detect around 250 genes that are highly specific for these T cells. So using these genetic signatures, we can look at them and we can annotate the different cell types based on this. So this is how we end up identifying and calling different T cell subsets. We are looking at the gene signatures and we try to identify them based on that. So let's look at how a T cell response looks like in an acute and chronic viral infection from some of our data. So here on this UMAP, again, I'm showing around 60,000 T cells from the acute and the chronic infection model that I referred to at the beginning. And here we are looking at clonally expanded T cells, which means that T cells are reacting to the virus. So first, I show you the acute infection model where the virus can be efficiently cleared. And you can see that at day 8, these blue cells, these are clonally expanding, so they are reacting to the virus, and they are found in this effector state. This is what you expect. Once the T cell is activated, it goes into this effector state, and it will clear the virus, it will kill virally infected cells or cancer cells. After this is a day 8 time point. So very early on, this is what is happening. At day 21, when the virus is almost completely cleared from the mouse, T cells that are clonally expanded can be found here on this UMAP. And these subsets are memory T cell subsets. So T cells already started to form memory by day 21. So what happens in the chronic infection model? At day 8, when T cells should make efficient effectors, they fail to do that. You can see that clonally expanded T cells that are reacting to the virus can be found in a different T cell state. We call these early effector exhausted T cells. They are not found in this classical effector state, but rather in this other exhausted effector state. If you look at the day 21 time point, then you ideally, by that time, you should form memory. This is what's happening. You don't see any clonally expanded T cells in the memory pool, but instead, you have multiple different T cell states where clonal expansion can happen. So the entire T cell response is derailed in this system. So something is happening to the T cells, they cannot clear the virus, and they acquire different T cell phenotypes or T cell states. So after this, we decided to look at T cell clones one by one, because what I'm showing you here is only T cells, all the T cells that are clonally expanded. But you can imagine, based on what I just said, when I was trying to discuss the T cell receptor repertoire, is that there are multiple T cell clones with different TCRs that can react to the same pathogen. So let's look at these T cell clones one by one. So in the acute infection model, if you look at these clones, each and every bar here is a clone, and on the y-axis you can see the number of cells that are present in that one clone. You can see that there's a really efficient clonal expansion. Why am I saying this? Because if you look at each and every clone, you see that there are multiple T cells that are part of that one clone. So this clone is a T cell clone with one T cell receptor, and it reacts to the virus and it multiplies in number. This one T cell clone has 300 cells in it. So these are probably one of the most efficient killers that you can see. In the chronic viral infection model, at day 8, you see clonal expansion too, but what you can notice is that the number of expanded cells in a clone are much less. It's like 3 times less than in the acute infection model. So somehow, clonal expansion is reduced in the chronic infection model. If you look at the acute setting again at day 21, when memory cells are formed, you can see that at the clonal level, if you look at individual clones, this is what happens. You have mainly memory T cells formed in these clones. If you look at the chronic infection model at day 21, you can see that these clones are quite messy in a way. So cells in these clones are pretty large numbers, so they are clonally expanded. So this first clone has around 2,000 T cells, but their states are very, very diverse. So you have multiple T cell states. It looks like the T cell doesn't really know what to do with the virus, so it adopts multiple different T cell fates. But interestingly, if you look at more carefully, you might be able to notice some differences in how T cells behave in a clone. And that's what we define in the paper, basically, that some of the T cell clones with a specific TCR will follow a very specific differentiation program. And some of these clones are divergent in nature, which means that they can either adopt this blue fate or blue T cell state or red T cell state. Red is terminal exhaustion. Blue is TXKLR subset, which is a more efficient killer cell. So not all the T cells are going into terminal exhaustion. Some of them might be able to retain their killing effector function. Also, if you look at these other clones, which are much smaller in size, so for example, in this one, you have approximately 200 cells. These T cells can only adopt this blue state, which is really interesting because it tells you that some of the TCRs basically seal the fate of the T cell. So if you are born with a TCR, your fate is basically determined at that point when the pathogen is around. And you also observe some other T cell clone behaviors, like this TX term biased clone behavior. So these clones can only go to exhaustion. No matter what happens, if they have this specific TCR, T cell receptor, they just go to exhaustion. So let's trace these clones one by one. So we looked at divergent clones, and this is what we proposed based on the results. So the T cells start in this early effector state at day 8, and then they can differentiate to this progenitor T cell, then intermediate exhausted T cell, and they can bifurcate into these 2 terminal fates. One is terminally exhausted, so these cells are really bad killer cells, bad effector cells. This other one is a really good effector cell. That's what we think, although we are investigating this further right now. There are terminal exhaustion bias clones, so these clones can only end up in terminal exhaustion. And we have this TXKLR bias clones, which is really interesting because these cells can only end up being TXKLR cells, which are efficient effector cells. So what we are basically saying with this is that even though you are inducing T-cell exhaustion, you might have some T-cell clones that can still function properly. Probably the number of these cells is just reduced, so they can efficiently participate in the immune response and eradicate the pathogen. So how do these T cells end up having so different gene expression programs is my next question. And in order to answer this question, we need to understand that we have approximately 25,000 genes to start with, and cells use different sets of genes. So here I'm showing a very dramatic example, because T cells and neurons, which are so different in their morphology and function, they are using the same 25,000 genes, but they're ending up using very specific sets of these genes, so not all of them at the same time. And that's the reason why they can become so different in function and in morphology too. So what is the reason for that? So our genes are basically only 2% of the entire genetic material. So there is this very, very large proportion of non-coding regions, which is 98% of the entire genetic material. And this non-coding genome is really important. For a long time, we thought that this might be just junk because it doesn't encode for any protein. No genes can be found there. Genes are all in the coding regions. And so we didn't really know what is the function or what was the function of the non-coding genome. But it turns out that cells are using different parts of the non-coding genome to activate very specific gene sets. And so in order to understand this better, we need to look at this picture and realize that the DNA is an extremely long polymer. It's almost 2 meters long in each and every cell. And the cell needs to come up with very efficient mechanisms to pack this extremely long polymer into the nuclear space, which is around 4 to 5 diameter, 4 to 5 micron in diameter. So how does the cell solve this problem? So there are these proteins called nucleosomes, and the DNA is wrapped around these nucleosomes. So this is the first level where the cell can compact this large polymer. And this is what we call chromatin. It's basically the DNA wrapped around these nucleosomes, which are proteins. So in this tightly spaced nucleosomal structure, there is no gene activity. So genes do not work from these regions of the genome. What you need to do in order to activate a gene is to remodel the chromatin state. So start from here and use very specific enzymes to basically kick out some of these nucleosomes and make the entire gene body accessible for enzymes like the RNA polymerase II. The RNA polymerase II can bind to the gene body and transcribe the DNA sequence into RNA. And this RNA is going to be used to make protein at the end, and the protein will change the function of our cell. So this is the coding region. This is where the gene is encoded. But there are also these non-coding genome and non-coding regulatory DNA regions that I was talking about. And I just want to name here two. One is the promoter region, which is a DNA sequence that can be found right upstream of the gene. And the other one is enhancer region, which is another DNA sequence that can be found very, very far away sometimes from the gene. But the interaction of the enhancer and the promoter is critical to drive gene expression. And the cells, in order to become T cells or neurons, they're using very, very different enhancer sets to activate specific genetic programs. So is there a technology to map these open chromatin regions? So open chromatin region can be, I forgot to mention this, open chromatin region is usually detected at genes that are active and promoters, and also at enhancer regions. So these places are nucleosome free, as you can see here on this cartoon, and proteins can bind there and activate genes. So there is this method that we use in the lab, which is called assay for transposase accessible chromatin, that can map these open chromatin regions in the entire genetic material. So again, going back to this cartoon, this is how it looks when the cell is basically folding this large polymer DNA into the nuclear space. And if you stretch it out, this is what you see. You have nucleosomes, DNA is wrapped around the nucleosomes. This is a closed chromatin state, so inaccessible. Nothing can access the genetic information there. And this is an open chromatin region, which is probably, it can be an enhancer, it can be a promoter, or it can be a gene that is active. So to map these open chromatin regions, we are using an enzyme that is called TM5. The TM5 enzyme can be supplied with two different adapter DNA sequences. Now, when we incubate TM5 with our cells, it goes into the nuclei of the cells, and it's going to preferentially bind to open chromatin regions. And it does not only bind there, it also cuts these regions and pastes these red and blue sequencing adapters, DNA sequences, to these open chromatin regions. We can then amplify these with a polynucleotide chain reaction, PCR. I'm sure you heard about this a lot nowadays, because this is also used to diagnose COVID. And then we can sequence these DNA regions. And once we look at their positions in the genome, we can see that these reads, these sequencing reads, are enriched at open chromatin regions. We can also do this at the single cell level, and we can identify exhausted T-cell subsets as well. So I'm going back to my favorite example, the clone 13 chronic infection model. And I just want to show you that single cell ataxic, so mapping chromatin accessibility or open chromatin regions throughout DNA material, you can also define cell types. Now, these are not necessarily happening, these open chromatin regions, on the gene body itself, but it can also happen at enhancer regions, promoter regions. So these are all non-coding parts of the genome. And if you look at some of these unique open chromatin regions that can be present on promoters on enhancers, you can find that there are a few thousand sites that define one T-cell subset. So you can use these as well to identify your T-cells. But how do you annotate these regions to the genes that they are regulating? That's the big question here. In order to understand this question better, we can look at the data representation or visualization, which is a genome browser snapshot of a genomic region which encodes for one gene. So this is going to be my example, PDCD1. So this is the reference genome that basically tells you the location of the gene. And here I'm comparing two T-cell subsets, naive T-cells that are not reacting to the virus, and exhausted T-cells that are reacting to the virus, but they undergo exhaustion because they cannot clear the virus. So if you look at the accessible genetic or genomic regions, you can see that it's highly different in the two T-cell subsets. So exhausted T-cells use these non-coding regions that are right upstream of the gene to drive the expression of the gene. Naive cells do not really use this, so they are not expressing this gene. The question is, how do you annotate these distant regions to the right gene? Because sometimes it's quite obvious that these are proximal accessible regions, so most probably these sites will drive the expression of this gene, but there are some really, really distant ones, for example, this one, which cannot be simply linked to the gene all the time. So how do you find these targets experimentally? There's another technology that we use in the lab, which can map 3D genome organization of the cells, and it's called microcapture-C. So it starts by cross-linking the cells with the chemicals. So what you're doing is you're making a snapshot of a cell. So once you add this cross-linking reagent, there's going to be no change in the nuclear architecture of the cell. You are fixing all the contacts. And if you look at this little cartoon here, you can imagine that, for example, this blue region might be one enhancer region. This green region can be a promoter region, and the gene is somewhere here. So let's imagine that. And there is this orange thing here, which represents a protein complex that holds together the enhancer and the promoter. And this is the conformation that leads to active gene expression. So now after this step, once we fix the cells and we have this state, we can use an enzyme called amylase, which is going to digest the DNA until it faces with a barrier. And this barrier is going to be the protein here that keeps together these distant DNA regions. After the digestion, we can perform something that is called proximity ligation. So we add another enzyme, which is called DNA ligase. And the DNA ligase is going to connect these ends here that are proximal to each other. It will preferentially do this because the protein is keeping these DNA sequences close to each other. And then the ligase is going to preferentially ligate these ones that are proximal to each other or close to each other. So now, since we have the reference genome and we know the promoter regions of the gene, which is really close to the transcription start site of a gene, we can design an oligo pool, a DNA oligo pool that can recognize promoters, specifically gene promoters. And usually how it works is, if you recall some of my previous slides, is that the enhancer is going to be looped to the promoter, and that is going to drive gene expression. So what we are interested in is what connects to the promoter. What are the distant genomic regions that can loop to the promoter and induce gene expression? So now if we use this promoter oligo pool, we can enrich for promoter-mediated contacts, and then we can sequence this library. So again, it ends in DNA sequencing. So I'm showing again an example. This is a genome browser view of another gene called perforin. This is what Tcells use to kill. And here we look at single-cell ataxic profile of this genomic region. So this will show you accessible non-coding regions that are far away from the gene. And the question is, are these actually contacting the gene promoter or the transcription start site of the gene or not? And so we perform microcapture-C, and here we are looking at the contacts that are enriched with the promoter region. So here is our viewpoint. This is the promoter. This is where the oligo hybridized. And then we can look at these signals, and the higher the signal is, it means the stronger the connection with the promoter is. And you can see that these interactions are lining up very well with these open chromatin regions here. So this basically helps us to understand these distant enhancers, where they are and whether they are connecting to the promoter and drive gene expression. So interestingly, we also have this one here in the middle, where we don't really see a strong interaction. So this is why we really need to do this experimentally. And we can end up with surprising observations like this, that sometimes open chromatin regions that are really far away in the non-coding genome can regulate gene expression. So this is what we think how it works in 3D. Obviously, we are doing much more sophisticated modeling thing, but this is just my drawing. So this could be the conformation, how this gene is working. So how do we test the function of these open chromatin regions? That's a different question, because even though you detect them as interacting partners of the promoter, it does not necessarily mean that they are critical for gene expression regulation. So this is going to be a different example. Here, this is a macrophage data set where we activate these macrophages with IL-4. So these are the control macrophages. These are IL-4 activated macrophages. And here, this is an open chromatin region, which is in question whether this is functional to drive the expression of the gene that is located here. So this is pretty far away. It's 11 kilobases down or upstream of the gene. You can see a different technology here that can map the position of RNA polymerase II, which is the enzyme that makes the mRNA from the DNA. And you can see that in control macrophages, there is no RNA polymerase II binding on the gene. In IL-4 activated macrophages, there's a lot of RNA polymerase II transcribing the gene. So this is indicative of the gene is being super active. Transcription is going on. So if we want to understand the function of these open chromatin region, we have to use CRISPR genome engineering, which is a method to specifically recruit this Cas9 nuclease into this specific site of the genome. We can achieve this by using a small RNA piece called guide RNA that can bind to any specific genomic region. We just need to design it the right way. We just need to give it a sequence that will recognize a genomic region, and it will bring there the Cas9 nuclease, and the nuclease is going to cut the DNA at that specific point, and we can disrupt the gene, or we can disrupt an enhancer region or open chromatin region, if you will. And this is what I'm showing here is a gene expression measurement of the gene. So here, what we are doing is we are measuring the mRNA that is produced from the gene. So in control macrophages, the gene is not active. If you activate the cells with IL-4, you can see that the gene is becoming highly, highly active. If you target Cas9 nuclease to this specific open chromatin region here, far away, and you cut the DNA at this point, you can significantly reduce the expression of this gene, which tells you that this distant regulatory region is indeed functional in driving the expression of this gene. So what are these technologies good for, and what's the major goal for us to use all of these? I think the ultimate goal for us is to be able to isolate T cells and engineer them in the flask. You can think about CRISPR engineering and reinfuse the T cells in a way that these T cells are going to be resistant to exhaustion, or they will be really, really efficient effector cells. And I think I've shown you a couple of examples today that we can definitely think about to improve T cell function. And one of these is to introduce very specific T cell receptors that will seal the fate of the T cell, and the T cell can only become a really effective killer machine. Or we can think about genome engineering. We can reveal these open chromatin regions, enhancers with single-cell ataxia. Then we can go on linking these genomic regions to each other by understanding the 3D genome conformation of a cell. And then we can disrupt these regulatory regions, which will affect the gene expression program of the cell, and the cell is going to be functional in a different way. And again, I'm going back to this again. By introducing a new T cell receptor, a new specificity can drive a T cell fate that is definitely wanted, and it will yield an effector cell that can clear the virus or cancer. And I think with these, I'm going to stop. I think it was a lot of information to digest. I just want to acknowledge to my beautiful colleagues. Obviously, this would have not been possible without them. I have many, many collaborators, and I'm really lucky to have all these people helping me and pushing the boundaries of T-cell research. Thank you very much for your attention, and I'm happy to take questions.
Please remember to unmute yourself, as I can mute you, but I cannot unmute you. Actually, to begin, I do have one question. If you think about the different kind of measurement devices, observation devices in the last 5 years, which one made the biggest difference?
I think definitely single-cell sequencing is an amazing cutting-edge technology that was recently developed. And I think using single-cell gives you the opportunity to uncover cell types that you have never seen before and we didn't even know exist. So definitely single cell is something to look after, because it just extends our opportunities in multiple different ways. The other technology that I would call your attention to is 3D genome mapping, because yeah, just as I referred to it, the DNA is a really long molecule, and the cells need to package this long polymer in a very efficient way so that they can mobilize very specific parts of this DNA when they need to and activate genes from that part of the DNA. And I think it's really important to understand how cells move around this long polymer inside the tiny nuclear space to function, basically.
Thank you.
Hi, my name is Laszlo. I have a question. Can you tell us how are these T cells related to the COVID and the mRNA based COVID vaccine? Are they related, or is there a connection? Like how does
yeah?
This
so I guess your question is about how these mRNA vaccines might activate T cells, right? I think that's
yes.
Mm-hmm. Yeah, so I can go back to this slide quickly. This one. Yeah, so once you receive your mRNA shot, the same thing is happening, or a very similar thing is happening to when you actually get infected by the virus. So these cells, these immune sentinel cells, macrophages and dendritic cells that are present in the tissue, even at the site of the injection, they can just take up these mRNA sequences and they express the spike protein in that case from the mRNA. So then the spike protein can be presented on this MHC molecule that I was talking about here. And then T cells with the T cell receptor that can recognize this spike protein will be activated. They undergo clonal expansion, so the right TCR containing T cells will be clonally expanded. They multiply in numbers, and then they will contract. So some of these T cells will die, and some of them will make it to this memory pool. And this is going to be really, really important, because once the T cells are in this memory pool, they are already trained, they are primed to respond to the virus. And so if you get infected by the virus at this point, these cells are going to be super efficient in killing the virus. They are going to be much faster to respond to the virus, and they will have a much more efficient cytotoxic program so that they can clear the virus.
Thank you. And so these like very effective T cells, they are they like, specific to each individual? So you can't just clone them and
Yes.
like create.
Yeah, so this is also another crazy fact about T cell biology, is that if you compare two individuals in terms of their TCR repertoire, which is 10 to the 19 different T cell receptors, you don't really see an overlap. But yet, these TCRs can react to the same pathogen.
And can they be transferred, or is it safe to transfer?
Yeah, so transferring can be a problem because you cannot just transfer between humans. You have to know whether these T cells are compatible. So it's similar to transferring an organ into another human. So you have to do a very careful evaluation before you would do the transfer, because it can induce this so-called graft versus host response, which is a really crazy immune reaction, and that can cause problems to the host by these increased inflammatory mediators that can cause a lot of harm. So you can actually die from it. It can cause something like a cytokine storm. So all of these inflammatory mediators are going to be there, and they will cause a lot of harm to the healthy tissues as well. So transferring them is not really a viable option here. What you can do is you can take someone's T cells, something that I've shown on one of the slides. You can culture them in a flask, you can modify them, and you can re-infuse them into the patient to help. But this is not applicable to COVID. It is applicable to cancer. And there are amazing results with this. So you can reprogram your T cells, you can re-infuse these T cells, and they are really efficient in killing cancer cells.
Thank you. And how widespread is that the treatment for cancer? Is it like very new cutting edge in just certain areas, or is it like spread to some countries already?
Yeah, so I think it's getting broader and broader. The problem is that it's really expensive to do, because you have to isolate these T cells, you have to culture them in sterile conditions. So you have to be really strict. And then you are making viruses that can actually reprogram your cells before the transfer. So I think it's definitely getting more and more accessible widely. But at this point, I think it's targeting a narrower patient set.
Csaba Halász has raised his hand.
Hi, thank you, Zoltan. So I have probably two questions. One was that if I understood correctly, even in the exhausted state, there are exhausted T cells, but there are also some T cells which are still can fight off the virus. So my first question would be if I understood that correctly, and if so, do we understand why the body is not producing more of the good T cells which will fight off the virus? And can we basically encourage the body to start producing more of the good T cells instead of taking them out in a flask and multiplying it outside of the body and then putting them back?
Yeah, so this is actually a good question. So there are these TXKLR cells that are more cytotoxic. That's what I've shown. Yes. So I think it definitely, as I said, it has to do with their TCR. So the T-cell receptor that is present on the surface basically determines their fate. And I think, yeah, by some sort of treatment, I guess that was your question, if there is any type of treatment that can make these T-cell clones to produce more of the good T-cells. Well, yeah, I don't know. I don't have an answer to that right now. What I know is that if you use, and I can go to this slide. So if you use these antibody treatments that are targeting some of these co-inhibitory receptors on the cell surface of exhausted T cells, you can kind of reinvigorate these exhausted T cells. So they are going back into a more functional state. And this is another immunotherapy application that is FDA approved. And there are two antibodies, one targeting this CTLA4 and the other one is targeting PD1 on the cell surface. And definitely that was also a big success. It was a Nobel Prize too. And if you use these antibodies, you can reprogram exhausted T cells to be better effector cells. I guess this is answering your question.
Partially. I was more like, if you go back, I think it was in a previous slide, there was a bifurcation where we went into exhausted T cells and then the blue ones, which were good T cells. I don't remember.
Mm-hmm.
And basically, this one, and then there was like basically a step diagram with some bifurcation.
Mm-hmm.
Anyways, so at that bifurcation, I was thinking here, yeah, exactly, top right hand corner, if there is a way to basically encourage the body to produce more of the blue good T cells. So your answer was really good. I didn't know about that, to basically try to reinvigorate the red guys. But I was more wondering, can the body produce more of the blue guys, the blue T cells?
Yeah, so since these T cell programs are actively shaped in the environment where they act, I can imagine that there are certain environmental signals that can push the cell down more on this route instead of this route. There's no specific example that I can tell you about this, but I can definitely imagine, because these cells are fully equipped with sensors all over their cell surface. So they sense where they are, they sense the environment. So if you might be able to think about a drug or something that can program them, maybe acting more on this green subset, and then making this green subset to make the decision to go down this TXKLR differentiation route instead of the terminally exhausted.
Got it. Thank you.
There's no specific answer right now for that, but that's a good question, yes. One thing that we are actually trying to do is to modify these cells. This is, again, similar to the example that I was talking about when you take out a patient's T cells and then modify them in the flask and then reinfuse them. So we are trying to take these cells out from the mouse at this point and then take out certain genes that can make them to choose this path, not this one. This is kind of an extension to my answer.
Laszlo has raised his hand.
Yeah, I was just wondering, what challenges do you have? What do you need to progress more? Like, are there any equipment that's missing, or like tools that are, if you could do this and that, then that would be much easier for you to make some more progress?
Well, yeah, the technology is rapidly advancing, I would say. We have insane tools now to study how certain genes act at these transitional points and what those genes do. How do these gene programs or genetic programs instruct the cell to differentiate this way or that way? But you know, that's actually a really good question and a hard question to answer. What do I need? I'm actually more on the basic research side. So I'm trying to tell people that there are different clones, for example, that behave differently in these infection settings. So this can definitely trigger some more future work. So for example, this specific result tells you that it really depends on the Tcell receptor, what Tcell program you have. And if you manage to collect Tcell receptors that give you this specific Tcell fate, then you're going to be a better killer. So modifying these cells in vitro and giving them back to the patient is really something that has a lot of bottlenecks. So for example, you need to have enough cells. You need to be very efficient when you are expanding the cells in vitro. And what we can use to modify T cells is a retrovirus. So the retrovirus is actually cutting the DNA of the cell. And no one really wants that, right? You don't want to reinfuse something to the patient that is genetically modified. That's something that you really want to avoid. And the FDA is not really a big fan of that, but some of these protocols are already accepted because it seemed to be safe, and you got really nice response in the clinic. But I think we are facing with an issue of how do you modify these cells genetically in a safer way. Viruses are good. They are much better in basic research in the lab. That's something that we are doing. In therapy, it's more difficult to use viruses because you cannot fully control them. So I would say that modifying these cells is definitely something that we need to improve on. And that's hopefully gonna yield better therapies.
So if we don't have more questions, then please join me in thanking Daniel for his very informative presentation. And I would like to invite everybody to go to our website at hungariansv.com if you would like to get notification about our upcoming events. We usually have once a month. Or also, if you have a presentation in mind you would like to give, please contact us and we can talk. And with that, thank you again, Daniel, for this great presentation, and hope to see everybody very soon.
Thank you very much.
Yeah, thank you, Daniel. This is Joseph.
Bye-bye.
It was amazing. Thank you. Bye, guys.